That would be good.
Okay. So I appreciate everyone joining us for Grand Rounds this month of April, and we are honored to have some of our peers from the who are working on microRNAs and neurodegenerative conditions. So again, an overlap in terms of our focus areas, but with a slightly different angle. And so this always gives us a chance to learn how others are approaching the similar populations, but studying slightly different items from a different way. So, Dr. Simpkins, and I'm going to give a little bit of their bio. Dr. Simpkins is the director of the Center for Basic Translational Stroke Research. You have a COBRA in stroke, right, Dr. Simpkins?
Yes.
COBRA grant. And those are particular focus areas on topics that are very pertinent to us here in West Virginia. Bringing in a group who looks at that particular topic, in this case stroke, to move that effort along. And so Dr. Simpkins is the director of that, as well as interim chair and professor of the Department of Physiology and Pharmacology. And then Elizabeth, or you go by Liz, right?
Yes.
Okay, Liz Engler. And then how do you pronounce your last name appropriately?
Chiurazzi.
Chiurazzi. Thank you very much.
You pronounced it
Dr.
wrong.
Chiurazzi is a research assistant professor, also within the Department of Neuroscience at West Virginia University. So we're going to learn a little bit about mRNAs. Dr. Simpkins and Chiurazzi, your audience is a wide variety in terms of what they might know about mRNAs. So I'll follow the chat, but feel free to bring us out of our shell.
I will. So are the slides now up on your screens?
They are.
Okay, great.
And the way you can advance them is you're usually page down, or on the left-hand side you might have a page number 1, 2, and you can...
Okay, do we have a background of microRNAs up right now?
Yes.
Okay, great. Let me start there, and I'll be comparatively brief. MicroRNAs are short, usually 22 nucleotide non-coding RNAs, which means that they do not code for any proteins, so their job is not to make proteins. But they are found packaged in small lipid vesicles called exosomes, and they are secreted by every cell in the body. Now, what's unique about microRNAs is that they complement with messenger RNAs, the RNAs that are coded for proteins. And when they do that, they either reduce the translation of those messenger RNAs into proteins, or they target the messenger RNAs for degradation. So in effect, microRNAs reduce translation of messenger RNA into proteins. Now, microRNAs can complement with messenger RNAs either completely or partially, and partially is the typical case where 4 or 5 up to 7 or 8 of the bases or nucleotides complement, and that's sufficient to inhibit protein synthesis. The other observation that's been made is that multiple microRNAs can interact with a single messenger RNA. Now, we became interested in microRNAs primarily in the Alzheimer's field, but then have expanded it to other areas that Liz will talk about, because microRNAs have the capacity to produce complex phenotypes like are seen in Alzheimer's disease, where you get plaques, tangles, synapse loss, mitochondrial dysfunction, etc. So we became interested in microRNAs. The other thing about microRNAs is that they're likely very much involved in cell-to-cell communication. So these exosomes into which microRNAs are placed, along with proteins, messenger RNAs, etc., can be and are secreted by the cells that make the exosomes. So they're little packages of lipids that are released by one cell, and they can affect neighboring cells. And in fact, you'll see some data that they show up in blood of human subjects and rodents. So they're likely involved in normal processes like development, but also in the spread of diseases, and we're particularly interested in how neurodegenerative diseases spread. Now, what's shown on the screen is, and you don't need to read the details because there's lots of information. With the Human Genome Project, you can now go in and ask questions about particular microRNAs and what messenger RNAs they may complement. So one we've become interested in, and I'll show you data that gives a rationale for our interest in just a bit, is microRNA 34A. And what we did is in searching...
I'm sorry, I think, Dr. Simpkins, I think that your slide, we're still on the title slide.
Oh, really?
Yes.
Okay. I advance them and they come up on the screen here. I don't know what else to do.
Okay. We have many ways to deal with this, let's say.
So we're on 1, 2, 3. We're on slide 4.
Okay, here we go. I'm going to do this, and you just tell me. I'll get to 4.
Okay. Do you want to run the slides?
Yes, I can do that.
Great. So go to slide 4.
Okay.
And can everyone see it? Okay. What's shown here is microRNA 34A complementing with a variety of messenger RNAs that are involved in synapse formation and function, oxidative phosphorylation, which is a mitochondrial function. I'd go back one slide. Glycolysis, which is another way of forming energy in the cell, and then other messenger RNAs that code for proteins that are very important in cell life, cell death, and longevity. So this is an example of one microRNA that's coding for, in this case, about 20 different messenger RNAs. So if this microRNA hits all these messenger RNAs, then it's certainly able to affect the function of the cells that are being affected by 34A. Next slide. So we obtained a variety of tissues from the Duke Brain Bank representing areas of the brain that are affected in Alzheimer's disease, particularly the temporal cortex and frontal cortex, and then areas that are relatively preserved, like the cerebellum. And from that tissue, we isolated the exosomes that contain the microRNAs, and then quantified the amount of microRNAs in Alzheimer's brain tissue versus cognitively normal brain tissue. Next slide. And these are the data. The upper row of panels is the temporal cortex, the middle is frontal cortex, and the bottom is cerebellum. What we noted in these Alzheimer's subjects was there was an overall increase in concentrations of 34A in exosomes derived from the brain. But equally interestingly, the amount of 34A tracked with Barak and Barak stage, which is a way post-mortem to stage the severity of the disease. So at least in the temporal cortex, 34A is tracking with how severe the disease is. We found no change in the frontal cortex and no change in the cerebellum. So that said, this microRNA may indeed be involved in the events in the temporal cortex in Alzheimer's disease. Next slide. So we also then looked at, evaluated proteins that we knew the message for which was complemented by 34A. And in these three categories of proteins, synaptic proteins, oxidative phosphorylation proteins, glycolytic proteins, there was a substantial reduction in the temporal lobe of all of those proteins. And on average, it was about a 50% reduction, indicating that 34A elevation certainly could be accounting for all or part of the reduced protein levels in the brains of these subjects. Next slide. And I think what made that even more interesting was in the cerebellum, which is not affected in Alzheimer's disease, we did not see these changes in protein levels. So it said something happening in the temporal cortex with 34A could be of interest. Next slide. So the next study we did was with a mouse model for Alzheimer's disease. And this is a mouse that has inserted into it human mutations for amyloid precursor protein, presenilin, and tau. And those three proteins are very much involved in plaques and tangles in the brain. And what we observed in this study was when animals are young, 3 and 6 months old, we saw no changes in levels of 34A in any of the regions that we evaluated, including hippocampus, temporal cortex, frontal cortex, and cerebellum. However, at 12 months, and this is the time at which the animals have pretty severe Alzheimer's-like brain problems, they have cognitive impairment, there was a substantial increase in 34A in hippocampus and temporal cortex, and no change in the cerebellum or the frontal cortex. So that says that 34A elevations are happening in relevant brain regions of animals. So again, this is a connection of 34A with Alzheimer's disease. Next slide. We then took nerve cells from rats and grew them in a dish, and then transfected into them, inserted into them the code for 34A, various concentrations of the code, and asked the question, what happens with mitochondrial protein? So the left panel shows an assessment of mitochondrial function, and every aspect of mitochondrial function that we looked at was severely suppressed when microRNA 34A was overexpressed. And as we had seen in Alzheimer's patients, levels of proteins that we know are critical to mitochondrial function, and these are electron transport proteins, were reduced by 50% to 70% when we caused the cells to make too much 34A. So that said, in a dish or in an animal or in a human, 34A overexpression has what we believe are negative effects on energy metabolism. Next slide. Now, based upon that, we made a transgenic mouse that had 34A, the DNA for 34A, hooked to what's called mCherry. It's just a red staining molecule so that we know we got effective transfection into the embryos of these animals, coupled to, it's called a TET-on, TET-off system, so that we can use a molecule like doxycycline to turn on the gene. So that allows animals then to develop into adulthood We turn on the gene, and then we can ask the question, what is the consequence of animals who are young, healthy, have no other problems of which we're aware, but then we very quickly elevate levels of 34A in those animals. Next slide. We looked at various tests for cognitive function or memory and cognition in these animals, and shown there are two tests that we used. One was a T maze, one was a Y maze. In both cases, the animals in which we had turned on the 34A gene were functioning about at random. That means they had an equal likelihood of making the right choice or the wrong choice, which is an indication that their memory is not any good. In comparison to the animals that are the same, except we had not turned on that gene. So within 2 months of turning on this gene, we've induced cognitive impairment in these animals. Next slide. So we asked the question, what was happening with other markers of Alzheimer's disease? Shown in the upper left is are what are called immunocytochemistry staining of brains from these transgenic animals that have the 34A gene turned on versus transgenic animals that don't have that gene turned on. And then the left column is the hippocampus and interinal cortex stain for phosphorylation sites on tau. Now, tau is a protein which, when it's hyperphosphorylated, it has too many phosphate units on it, it fibrillarizes, becomes dysfunctional, and has bad effects in neurons. And as you can see in the hippocampus, there's a great deal of hyperphosphorylated tau. The same is true of the interinal cortex, which is another Alzheimer's relevant brain area. And we don't see this when the gene is not turned on. So these animals are otherwise healthy relative to their tau levels. And if we look at the proteins in a different way, that is, we obtain the brains, grind them up, and then do what's called a Western blot analysis, we see a substantial increase in phosphorylation of this protein. Next slide. And this is another Western blot with another antibody directed at different sites on the tau protein. And it says that at various points in the tau molecule, there's too much phosphorylation going on. So this is an indication that the animals have a neuropathology consistent with Alzheimer's disease. Next slide. Mechanistically, this is happening because we're turning off a molecule that promotes the process of taking phosphates off of tau, and we're driving it down to a very minimum level. So the protein gets a phosphate unit gets added, gets phosphorylated, and it's not able to be dephosphorylated or unphosphorylated, so it appears to build up. Next slide. Next slide, please. Okay. This slide shows cortex and hippocampus from those same animals, but now stained for an antibody that's directed against beta amyloid protein. And as you can see, both in the hippocampus, lower right, and cerebral cortex, upper right, there's extensive beta amyloid accumulation in those brains. If we haven't turned on the 34A, then we don't get that accumulation. The little bit of staining that you see in the left panel is probably non-specific staining because of the dyes used. So we produced a second hallmark of Alzheimer's disease. That is beta amyloid accumulation. Next slide. The mechanism by which overexpressing this microRNA in these mice causes beta amyloid accumulation could either be because it enhances BACE1, that's a beta secretase, that drives the pathway towards A beta production, or it could be because of the suppression of a non-amyloidogenic pathway, that's alpha secretase. And so we probed that to answer what was going on when 34A was overexpressed. And we had known through these in silico analysis that ADAM10, which is part of alpha secretase, the non-amyloidogenic pathway, was the target of 34A. So we assessed ADAM10, next slide, and saw a profound reduction in ADAM10 in various areas of the brain when we overexpressed 34A. Next slide. This will be my final slide, and Liz can take over. So 34A, as well as another microRNA that we've looked at, but I haven't shown you any data for, are overexpressed. Too much is being made in Alzheimer's disease. They compromise brain bioenergetics. They stimulate tau hyperphosphorylation, which is the precursor event to neurofibrillary tangle formation. And they cause the accumulation of beta amyloid. So, and as I showed you, there's cognitive decline in these animals. So we think this animal model may be a good animal model for what's happening in Alzheimer's disease. And likewise, we think that microRNA 34A may be a good target for drug discovery for a therapeutic for Alzheimer's disease. So I will stop, and I'll turn it over to Liz to do the rest of the presentation.
Okay, next slide, please. Okay, so we're going to switch gears a little bit to talk about another example of an age-related brain disease condition, stroke. And I don't have to make a strong case for the worthiness of studying stroke, especially in Appalachia, where we shoulder a disproportionate burden of this disease, given the hundreds of thousands of people every year that are affected by a stroke, and the many millions more who live for many decades with the debilitating consequences of this brain injury. So what we were interested in that may not be as well known is that stroke is a strong risk factor for the development of Alzheimer's-like dementia, and so investigation into the potential role of microRNAs, especially considering our observations in the Alzheimer's disease brain, and as well in the model of Alzheimer's disease that Dr. Simpkins just presented, was particularly important to us. And so to start this process out, we obtained blood samples from 13 patients who had been diagnosed with ischemic stroke and measured the levels of microRNAs in those samples. Next slide, please. And so what we find is that microRNA, as we saw in the brain and blood of Alzheimer's disease patients and in rodent models of Alzheimer's disease, in stroke patient human subjects, we see that blood levels of microRNA 34A are persistently elevated during not only the acute injury period of the 6 to 24-hour range, but in the subacute period, 5 days out, and then more long-term, looking at our 30 and 90-day time points towards the far right side of the graph. Okay, next slide. So work by a colleague of mine in the Simpkins laboratory, Dr. Sophie Wren, took the next step in translating this back to rodent models of ischemic stroke. And the way that we study stroke in rodents is to induce an experimental stroke by exposing the carotid artery of anesthetized animals and feeding into it a filament that blocks the flow of blood into the middle cerebral artery and into the brain. And we leave the filament there for a set period of time before we remove it so that we can model the reperfusion injury that happens when blood flow is restored, like what we see in the human population when the chief FDA-approved pharmacological intervention, tissue plasminogen activator, is administered, or when the clot that is blocking blood flow to a human's brain is removed with mechanical techniques. So usually we do this in rodents for a 1 hour time point, and what you can see here on this figure is that in the serum of mice who have experienced a transient middle cerebral artery occlusion, that's the blue bars, there's a significant increase in the expression of microRNA 34A. Next slide, please. We also then took the next step to look at the consequence of deficiency in microRNA 34A in the context of experimental stroke. And so we bred animals to be deficient in this microRNA genetically from birth. And what we see, if I can call your attention to section C of this figure, is that when comparing coronal brain slices of mice that have been depleted of 34A to their wild type controls, what we find is a significant protection from the infarct that is induced by an experimental stroke. So in looking at the brain slices, these slices have been stained with a compound called TTC. This compound binds to living and thriving neurons and does not bind to neurons that are either dead or necrotic and close to dying. And so the white sections are examples of areas in this particular mouse's brain that were susceptible to the detrimental consequences of the ischemic stroke that we induced. And what you can tell just by looking is that the white areas are very large in the wild type left panel animal, but much smaller in the 34A knockout animal just on the right. And then in D, we have a quantification of the volume of those areas, and you can again see that the 34A knockout animals are showing a lower amount of infarcted tissue compared to the wild type animals. Next slide, please. Okay, so we took another step to try and demonstrate this phenomenon in the opposite way, by overexpressing 34A. So using a very similar animal strain to the one that Dr. Simpkins described a few slides ago, we bred animals to be conditionally overexpressing of 34A when we administered doxycycline. We administered doxycycline in the drinking water about a month before we induced a transient middle cerebral artery occlusion. And what you can find, again looking from the TTC staining, is that the wild type control animals show some level of infarction. They show some white discoloration of their brains following a stroke. But the animals that had 34A inducibly overexpressed in the weeks prior to their stroke show a much higher infarct volume in various brain regions, and as evidenced by a much larger white region of their brain. This also corresponds to significantly elevated neurological deficits in the acute injury period, as demonstrated by the bottom left graph. So we're measuring for whether animals are able to walk appropriately or are circling or falling down to the injured side, and demonstrating some level of paralysis. And then the right graph towards the bottom of the screen just verifies that we indeed did get a significant elevation of 34A. So these effects on infarct volume and neurological deficit are likely attributed to the substantial increase in 34A expression that we achieved with this method. Next slide. So just to quickly conclude what we are seeing from the findings in the context of stroke, we know that from evaluating human subject and mouse tissues that 34A is overexpressed following the experience of a stroke. And what I didn't have time to present to you today, but that we've made observations for in other studies in the lab, is that this overexpression, as Dr. Simpkins alluded to, compromises brain bioenergetics, and it seems to significantly correlate with a worsened stroke outcome when this happens, and it seems to be particularly problematic for endothelial cells that line what's called the blood-brain barrier. Now, this is a barrier that separates central nervous system tissues from that of the periphery, and when it's intact, everything goes well in our body, but when it becomes permeable, like after a stroke, tissues and cells and secretory factors from the periphery are able to get into the brain, sometimes for beneficial consequences, but oftentimes exerting some pretty negative outcomes that, or negative effects that can really exacerbate negative outcomes in the future. So just to quickly summarize, it seems like deficiency in microRNA 34A is a protective feature in stroke, and that microRNA 34A overexpression can exacerbate stroke outcomes. So again, this highlights the potential role that this microRNA may have as a therapeutic target for novel interventions and possibly as a biomarker for the indication of a stroke-related injury. Next slide, please. Okay, so we'll switch gears one more time now to talk a bit about another type of acute brain injury, which is traumatic brain injury. So this type of injury has been traditionally associated with a young adult population where they're engaging in risky behaviors and fall or crash and hurt their heads because of a concussion or things along those lines. However, we are seeing that as the baby boomer population ages, traumatic brain injuries are becoming increasingly associated with a geriatric population that is becoming at increased risk for injury-inducing falls. And so understanding the mechanisms that control the progression of the disease will become particularly important as we develop novel therapeutic interventions that are useful for the general population of brain-injured individuals, and begin to understand what unique risks and consequences injury in an aging group confers. So to take a first preliminary step towards this, in collaboration with Dr. Jane Wigginton from UT Southwestern, we acquired blood samples taken from patients who had been diagnosed either with a traumatic brain injury or a brain injury as the result of hemorrhagic shock. So these were people who sustained a wound and lost a substantial amount of blood, thereby leading to a deficiency in brain oxygenation and therefore a brain injury from that. And we measure the microRNAs, a large panel of microRNAs that address a variety of cellular functions ranging from synaptic function and plasticity, immune response, and cell bioenergetics, just to name a few. Next slide, please. Just to give you a quick perspective of the inclusion criteria, this was a sample of subjects from a much larger trial that was investigating a few more endpoints, but I just wanted to highlight that subjects who were grouped into the traumatic brain injury arm could not also have had an injury where they lost a significant amount of blood, and vice versa. Patients in the hemorrhagic shock arm could not have had an injury that resulted in blunt trauma directly to the head. Next slide, please. Okay, so to show you a preliminary look at what we're seeing, we evaluated the panel of microRNAs that I just alluded to at multiple time points across the acute injury period, ranging from 1 hour post-injury all the way out to about 120 hours post-injury in both the hemorrhagic shock groups, which is the graph on the top of your screen, and traumatic brain injury patients, which is the graph on the bottom of the screen. This is a busy couple of graphs, so I'll walk you through it. Basically, the takeaway is that we can see distinct patterns of microRNA changes that are unique to a specific microRNA. They change across time, and they are potentially specific to an injury subtype. Now, I know that's sufficiently vague to be almost useless, but there are a couple of patterns that we can glean from looking at these two figures together. So the first pattern I want you to attend to is that in the hemorrhagic shock patients, if we're looking at expression changes across time of the microRNA panel as a whole, although we see a couple of microRNAs that spike early on in the injury period, around 6 to 8 hours, by and large, many of the microRNAs decline fairly rapidly in that early period and stay low throughout that acute injury time frame. When we look conversely at traumatic brain injury, we see more of a phasic change across time, and the magnitude of those changes appears to be much larger than in the hemorrhagic shock injury. The other thing that I want to call attention to is that there are some microRNAs, namely the sort of yellow-looking line 132, that seem to go up in the early acute injury periods of both traumatic brain injury and hemorrhagic shock. This may be a more global marker of brain injury rather than a specific marker of a certain injury type. And this is important because most of the literature evaluating changes in microRNAs compares a brain injured person back to a healthy control. And so this precludes the inclusion not only of healthy controls as your comparison groups, but also of control groups who have sustained some kind of injury, like a broken leg or a broken arm, that doesn't involve the head, so that we can get a sense of which microRNAs are injury specific to the type of injury we want to measure. And then just to call attention to microRNA 34A, it does seem like in both hemorrhagic shock and TBI, it follows a similar trajectory to the rest of the microRNAs, and it's kind of the middle of the line. And I just want to point out, again, with TBI, we see more of a phasic change where levels of microRNAs generally go up and go down, cycling about once every day throughout the injury period. And then again, we have it out to 120 hours. We don't know exactly what happens beyond that. But for the most part, this is one of the first studies to comprehensively assess the changing panel of microRNAs across time in such granular detail during this acute injury period. Next slide, please. Okay, so the conclusions here are mainly that there may be different mechanisms of brain injury that result in differential patterns of microRNA expression across the injury period. And one thing that I want to call attention to that we noticed is that this may be explained by the differential pattern in the treatment that is applied to each of these different injuries. So a common practice in the context of hemorrhagic shock upon arrival at the ER is that these patients receive blood transfusions fairly rapidly in the course of their disease, their injury treatment, because they've lost a substantial amount of their blood volume. And what this might be doing is flushing out the changing pattern of microRNAs that may lead to long-term pathological outcomes. In contrast, traumatic brain injury is very seldom treated with blood transfusions unless there's another type of need indicated. And that may explain why we see a much more phasic pattern of expression in these patients than we do in the hemorrhagic shock. And points to the possibility that blood transfusion may be in and of itself a therapeutic intervention to attenuate the pathological consequences of the disease progression. And then what we see is within a given brain injury type, levels of microRNAs are not static, but they are changing, and they are changing potentially dramatically and often across time. And so this precludes an important recommendation to the field, not just to sample at 1 day and then 5 days and 30 days wherever possible, but to try and really capture the potential variable changes in microRNAs during this very critical time post-injury. And lastly, that the pattern of change can be microRNA specific, but that's a good thing because it highlights the utility of specific microRNAs as either biomarkers or novel therapeutic targets. Next slide, please. Okay. So we've talked about a lot of different investigations that the lab is taking, and you can see that there is a truckload of data here, and there's an even bigger truckload of work still to be done. We are in the process right now in the context of the 34A and Alzheimer's disease connection of writing up an R01 grant application to the National Institute on Aging to more comprehensively characterize this novel animal model and to determine some important connections between miR34A overexpression and how it eventually dysregulates hallmarks of Alzheimer's disease pathology like beta amyloid and tauopathy, as well as cognitive decline. And we think the way that this is happening is through dysregulation of brain bioenergetics that's mediated by mitochondria. In the context of stroke, we also want to investigate the role that suppression of mitochondrial activity by 34A has in that condition, and we'd like to more thoroughly investigate how 34A is impacting the maintenance of the blood-brain barrier in the context of an acute injury like stroke. And finally, in the context of TBI, I showed you very preliminary data. We have a lot more work to do there, looking at the functional significance of the expression of these different microRNA targets across unique diseases and during the injury period, both acutely as well as more long-term. And the end goal of all of this work is ultimately to try to develop small molecule pharmacological interventions that reduce 34A activity, and the hope there is that we can exert beneficial outcomes on many disease states associated with aging. Next slide, please. I just want to end by acknowledging, I get the best part of the presentation, acknowledging the hardworking efforts of a very collaborative and expansive team of scientists. So I want to call special attention to Dr. Sophie Ren and her husband, Dr. Hang Hu. They are pictured right near the front, right behind Dr. Simpkins. And their work was instrumental in the connection between microRNA 34A and stroke. Just to the right of Sophie is Dr. Shoman Sarkar. His efforts were key in developing the Alzheimer's disease and 34A project line, and then a variety of other people, including Sarah Lewis, Sujung Jung, and Sarah Naeem, an enterprising and hardworking undergraduate honors thesis student who's not pictured, as well as myself on the TBI project. And I'll also acknowledge the funding sources that we have that paid for this work, and we can take any questions.
I'm just back so that I can look at any questions.
Can you hear me?
A little louder, please.
Hi, I'm Charlene, and I work with the Traumatic Brain Injury Program.
Uh-huh.
And I really appreciate all that you all have shared today. And I actually have to say I have a lot of hope for what you are researching for folks with traumatic brain injury, but also for Alzheimer's disease. It sounds wonderful.
Thank you.
Thank you for all you're doing.
So in terms of, while people think about a little bit more in terms of on the chat function, and I don't know, Dr. Simpkins, if you guys can see the chat room, but if not, I can mention some of these. Just help us. What's the average timeline from work that you do to potential medication? What is that average timeline, would you guess? I know it differs by condition and circumstances, but that translation is very important, and I'm not sure if everyone.
Yeah, I in general, to go from preclinical observations to clinical studies is probably 3 to 5 years, and then in the clinic it can be as long as a decade to do the phase 1 safety in human studies, phase 2 initial efficacy studies, and then phase 3, which are the larger population studies, can be a decade or longer. And I think it depends upon the condition, it depends upon the quality of the preclinical data, and obviously depends upon what is observed in the various phases of the clinical study. So it is not a rapid process, but it's one that we want to undertake. Liz alluded to one that may go actually fairly quickly. We've observed, Sophie Wren has a project in which she does blood transfusions after stroke. And she's found that if a blood transfusion is done out to 11 hours after the onset of stroke, she gets very robust protection of the brain. So on the basic science side, we're trying to work out what part of blood is involved in that protective effect. And we have leads on that. We're continuing to pursue that. She's has submitted grant applications to fund that. But we're talking to the neurologist and the hematologist about starting an initial clinical study of transfusion either of blood or a particular blood cell type to see if we get benefits in stroke. So we're excited about that. So in that case, it would have been about 2 years from initiating this project preclinically in animals to getting to a point where we do an initial assessment in human subjects.
Okay. Thank you. I think from a couple of different perspectives, just so to give you a little bit more about your audience, you heard from Charlene, Miranda as well on our TBI team. They, of course, have the TBI registry, so every event that happens in West Virginia is reported, and then they follow up with a survey on particular characteristics of the event, noting intensity, location, and then follow-up services. You mentioned in your past study pretty immediate follow-up with the individual. We'd have a variety of people even with the event happening a while ago, but in terms of their potential role in collaborations with you, that's just something on their end that they're doing, and they're providing services to those individuals, whether they received immediate services and are still having issues or not, and that's across the age groups. And then you have a wide variety of programs represented on here for people who are attending, who, again, lifespan, right, different age groups, who might be more susceptible in terms of stroke and Alzheimer's, and again, kind of their role and ways in which they might be helpful to you because they are interacting and providing services with individuals directly. So just as an FYI, that's helpful from your end in this process at large.
Sure. I can see a number of areas, and Liz, you can answer too, in which we may want to collaborate, since we're, molecular biologists, biochemists, pharmacologists. It would be interesting, for example, for us to know in your TBI population, groups of subjects who go on to show cognitive decline versus those who are very resilient, recover, and don't have a remarkable change in cognition, to try to characterize microRNAs or other things in the people who progress to cognition versus groups that don't progress. An area, and I'll let you talk about your post-stroke depression studies, but I'll turn it over to you.
Sure. So one area that I'm particularly interested in is post-stroke, but more broadly even post-brain injury depression, which is a pretty pervasive problem among people who've experienced a brain injury, as I'm sure you're aware. And it happens at much higher rates than the population. And of course, there's the obvious conclusion that exposure to a life-altering brain injury is going to be associated with some struggles and some emotional disruption. But I and many others firmly believe that there's also a pathological cascade that's happening as a result of the injury that may be leading to the development of depression that is particularly resistant to effective treatments. And so having access to, even just first of all, getting a clear sense of what the rates of these secondary consequences like depression are in these populations is a huge step, because often what we find is that rates of these complications are grossly underreported because people are lost to follow-up. They, especially here in West Virginia, experience their injury, come to Ruby, get their treatment, and go back home, however far away or difficult to travel to that is, and we simply never hear from them again. And so certainly just getting a sense of what the scale of the problem is accurately in Appalachia and more broadly would be a significant help to the field.
That's great. I know our TBI, and Charlene and Miranda, please feel free to jump in. You're working with these individuals definitely directly, but I think those issues, particularly depression, is something that our team has expressed. That is a significant issue, and have really started to develop or gather tools to work with the individual on that piece. Substance abuse goes hand in hand with that, and that's another one that falls pretty shortly behind. The other thing, and I don't know if this is of an interest, but there are other individuals where it's not just one TBI event, it's multiple TBIs. So when you have that multiple impact, particularly in different areas of the brain or maybe in the same area, how do they look differently from ones that might have one, and then of course your controls?
Yeah, very good. Just to follow up on Liz's comment, Liz has very good preclinical evidence that a defect in the immune system may be predicting the development of depression, whether it's spontaneous depression, like in major depressive disorder, or an event-related depression. And so she's working on that in a preclinical model, but that's very amenable to assessing in a patient population. Mm-hmm. So it's, I think there's a number of ways that we could interact. And I certainly would like to have follow-up discussion about that. And Miranda, you had asked if you can have a copy of the presentation. Yeah, Leslie, you're welcome to send that presentation to anyone who you like.
Okay, thank you. And Miranda is our program manager for the
Great.
BI. So fantastic. So with that, any other questions? We're at 3, a little after, but I don't want to, any other? And I want to thank Nick Larson. Nick Larson was he's a member of our Positive Behavior Support Program, and he told us about your work and research and offered the suggestions. So thank you, Nick. I think this is wonderful. Appreciate it.
Good. Well, thanks for allowing us to present this work to you. Appreciate it.
Absolutely. I'll follow up with you with the connection to the TBI group so that can continue, and I appreciate that.
Great, thank you.
That would be very good. Thank you very much, all of you.
Thanks for having us.
Thank you.
Thank you. Have a good day.
Okay, you too.